Ladies and gentlemen, Your Royal Highnesses, dear Jonk Fuerscherin, it is with great pleasure that we welcome all of you today to the fourth event of the Science Next program, Water Futures. I'm Özgü, a former laureate of the Jonk Fuerscherin contest myself, and I'm a current science student at the University of Luxembourg. And I represented Luxembourg in the European Union contest for young scientists in 2018. And therefore, today I'm very happy to be here again with the Jonk Fuerscherin. I will be moderating the conference this afternoon, and for technical reasons, we will be having the conference in English, but please feel free to stop me at any moment and ask any questions that you might have, and if you don't understand what is meant or said. So we'll all help and translate as best as we can. And at the end of the conference, you can ask any of your questions in Luxembourgish, English, French, or German. And yeah, so stellt Frauen op Luxembourgisch, Deutsch oder Französisch. So please don't be shy. Considering the difficult circumstances that we find ourselves in with the ongoing pandemic for the past 2 years, I feel very privileged to be here today on this occasion. And for that, I think we owe gratitude to scientists and those who support scientists. However, the global health crisis that we've been battling with as mankind did not hit pause for the growing concerns regarding other issues on a global scale, such as loss of biodiversity, climate change, rise in sea levels, water pollution, and ocean acidification that we're all familiar with. And to be honest, these issues all seem to be interconnected and can be traced back to increasing demand for an unsustainable and unstable management of non-renewable resources. So before I hand over the microphone to our scientists, I would like to invite Carlo Hansen, the president of the Fondation Jeune Scientifique Luxembourg. And Carlo has been supporting research and science for over the past 20 years, and he's been the president of the foundation for the past 14 years. So I would like to please invite him with a big round of applause to the stage.
Your Royal Highnesses, dear young scientists, distinguished guests, welcome, good afternoon. Your Royal Highnesses, we are honored by your presence today, but also thankful for your and Princess Stephanie's interest and encouragement. Thank you all for joining us today. I am extremely pleased to welcome Mrs. Mara Manieri, a former Jonk Fuerscherin laureate, and of course, Dr. Laurent Pfister. Thank you both for being here today. Mara participated in the National Jonk Fuerscherin contest in 2020. For her science project, she took water samples from Luxembourg's rivers and streams and analyzed the impact of fertilizers and manure on the water quality. She was awarded in 2020 the participation at Europe's largest science contest, the European Union Contest for Young Scientists, organized by the European Commission. She is co-founder of a group called Change in the Lycée Classique de Diekirch, a group which aims at raising students' awareness to environmental issues, to sustainability, and how to protect environment. She also participated at the Luxembourg Biologies Olympiade. Dr. Laurent Pfister is head of the research unit Environmental Sensing and Modeling called ENVISION at the Luxembourg Institute of Science and Technology. He studied at the Université de Strasbourg and has a PhD in hydrology and water resources and is accredited to supervise research. He is a lecturer at the University of Luxembourg, author of numerous publications and associate editor of science journals. He's a group leader of a research team, the Catchment and Eco-Hydrology, which focuses on water catchments, water collection, storage, mixing and release, using to this experimental hydrology, like for example, biological tracers or infrared imagery. As you see, Mrs. Manieri and Dr. Pfister have a common scientific interest for one of Earth's most valuable resources, water. Water concerns all of us, and its management arises a lot of questions. How do we protect and guarantee its quality? How do we ensure that everyone has access to it or keeps access to clean and potable water? Its management and protection requires that everyone takes a real interest in the issues at hand: droughts, floods, modification of water quality, and the loss of biodiversity it entails. Remember, last year's floods in July, Echternach was partially down under, as well as the Province de Luxembourg or the Ahrtal. So we should care about water, a crucial resource we have the responsibility to protect and manage wisely. Everyone can contribute to do this, and I'm very proud to say that each year there are young people who present a scientific project at our annual national contest, which precisely aims the protection of our planet's resources. The National Jonk Fuerscherin contest, which exists since 1971, is not a talent show. It's a think tank, a tool for young people who want to make a difference by contributing through their projects to search for solutions, for answers, and to tackle those global issues, or at least explore possibilities how to solve these global issues we must face. Thus, the Science Next program our foundation offers since last year, thanks to one of our sponsors, is to be understood as an additional tool for all those who want to participate at the contest or intend to do so. The workshops and the conferences offered in Science Next program all aim at helping to reflect on issues, at documenting an observation, at doing a survey or an experiment or an invention, which brings us further and benefits all. So that's the gist of the Jonk Fuerscherin National Contest and the Science Next program, which goes with it. Enjoy the conference, and I give the floor to Mrs. Susanna Young. Thank you very much.
Thank you so much, dear Carlo. Your Royal Highnesses, distinguished guests, dear Jonk Fuerscherin, we are extremely grateful to welcome all of you for the opening of the fourth event of our brand new program called Science Next. So before starting, may I deliver some more information about Science Next, which is hosting actually the Water Futures Conference tonight. Thanks to our sponsors, Science Next has been launched on November 2021 and is since accessible for free at the benefit of all registered students in Luxembourg aged between 11 and 21. Composed of an extracurricular training cycle throughout the school year, Science Next provides with a syllabus that guides our young scientific enthusiasts from a starting idea of a project up to a public presentation of an scientific achievement, as it happens tonight with the coming conference. Ever since, some of our students I see present tonight, You have already learned about how to transform a simple idea into a successful project, how to make proper research on a specific topic, and how to structure the ideas into an impactful presentation. These tools are acquired by you, our young students, on different occasions at focus places where you actually meet real experts and scientists, such as Paul Wurtinkup Science Center, Luxembourg Institute of Science and Technology, and so on. But most importantly, Science Next is also a place to have fun, right? We had so much fun together. And not to mention that Science Next is also a place to be connected with your peers. So after all these wonderful experiences, today you will have the opportunity to see an example on how to present your project in a scientific manner. By offering this conference, Science Next is willing to show, based upon the example of our dear former laureate, Mara Manieri, but also Dr. Laurent Pfister, that your personal reflections can really contribute to the current challenges of our planet, and that you can really make the difference. Last but not least, I really need to address many thanks to our sponsors, to the board member of the FGSL, to Mr. Serge Quazzotti, the president of the jury of the contest, and a special recognition goes to Mr. Sébastien Wirtz. Thank you all for your incredible dedication, not only to this platform, but far beyond. Thank you so much, and enjoy the conference.
Thank you, Susanna, for this introduction. So, as Carlo had mentioned, now we will welcome Dr. Laurent Pfister and Mara to provide us with their outlook of different generations of scientists on the same topic, water, of course. They will develop on innovative instruments and protocols and offer environmental data with unprecedented temporal and spatial resolutions, which provide brand new information on how climate change is intensifying the hydrological cycle and signals in flood records. Now, I would like to invite Mara to the stage to hear about her outlook and presentation of her research. Please, everyone, welcome Mara with a big round of applause.
Hello, my name is Mara Manieri. Until last summer, I was a student of the Lycée Classique de Diekirch in the north of the country, and I'm now studying medicine in Munich. In 2020, I participated in the Jonk Fuerscherin contest with a project realized in 2019. I am here this evening to talk about the project, the competition, and what followed. The project is entitled Water Quality in Luxembourg and its Effects on Aquatic Microorganisms. I live in a rural area, and so I have kind of always wondered about the effects of excrements on the water in rivers and streams, but also on the microorganisms living there. And when I learned about La Fondation Jeune Scientifique and the competition, there was an opportunity to search for answers. I wanted to begin with some preliminary explanations. So the majority of terrestrial gases are nitrogen. This element is necessary for the formation of organic molecules that are essential for life on Earth. I'm sorry. I think we have to switch microphones. Yes? So, yes. I'm very sorry for the technical problem. So the majority of terrestrial gases are nitrogen. This element is necessary for the formation of organic molecules that are essential for life on Earth, such as proteins. However, most organisms can't directly use the atmospheric nitrogen. But we also find it under different forms, such as organic nitrogenous molecules. Via several processes, these can be transformed into nitrate NO2 and nitrate NO3. These two ions are very soluble in water. The organic nitrogenous molecules come from dead organisms and from animal excretions and feces. Naturally, the concentration of nitrates and nitrates in water is relatively low, and so without human intervention, they constitute a limiting element for plant growth and development. Phosphorus is another essential component of life. It can be found in nature as phosphate anions, PO4. Animals release the phosphate when their organisms break down, so after their death, or through their excretions and feces. The phosphate is relatively scarce in soils and consists therefore a limiting element for plants' growth and development as well. To increase the amount of nitrogen and phosphate available, nitrogen and phosphorus fertilizers are used in agriculture. They are then no longer a limiting element for plants' growth and development, but stimulate the development of cultivated plants. These numbers were the data I found during the preliminary research in 2019. The latest official StatTech statistics on fertilizer use in Luxembourg date from 2010. However, these figures do not consider organic fertilizers, manure, or green manures, which are nevertheless very widely used, especially by farmers with animal farms. So the real volume of nitrogen and phosphorus fertilizers is still much higher. There are quite strict regulations for the use of nitrogen fertilizers, both mineral and organic. Minimum distances to water courses and wells have to be observed. And these are greater for organic fertilizers than mineral ones. The use is also subjected to the weather. And finally, it is forbidden to apply fertilizers to the fields between the 15th of November, 15th of February. The regulations, however, vary depending on the areas under consideration. Water protection areas, such as around the Lac La Haute-Sur, are subjected to stricter regulations. The geoportal also indicates information on the state of rivers. They indicate an average concentration and the maximum concentration measured. However, this does not make it possible to follow the evolution of the different concentrations over the year and to conclude on the impact of agriculture. Moreover, at the time of my research, the latest published figures dated from 2017. So, to put it plainly, in some parts of the country, it is forbidden to put manure on the fields, as well as during certain periods of the year, especially from the 15th of November to the 15th of February. But regulations on fertilizer use are not an end in itself. They should seek to preserve water quality. So I thought that we should recognize, by analyzing the water, if we are in a fertilizer ban period or not, just as we should recognize if we are in a water protection area or not. But why do we seek to preserve water quality by fertilizer regulations? To allow aquatic life, not to hinder the proper functioning of the ecosystems. But what does exactly happen then when the water quality decreases? How do the organisms react? Do they vary in number? Do they vary in species? Generally, I thought that we should expect a decrease in the number of microorganisms observed when the water quality decreases. And just a little explanation, in this project, I define good water quality as low concentrations of nitrates, and phosphates, whereas bad water quality equals a high concentration of nitrates, and phosphates. In reality, it's naturally more complex than that. So the project itself consists of two different parts. The first part is about water quality of the rivers, and the second one is about the impact of bad water quality to aquatic microorganisms. For the first part, I took samples from different rivers across the country, and the exact locations are shown on the map. I tested these water samples for the presence and, where applicable, the concentration of nitrates, and phosphates, these by aquarium tests. And for the second part of the project, different amounts of slurry were added to water sampled from a pond. 24 hours afterwards, the water was analyzed under the microscope and the microorganisms were counted in a hemocytometer. And in order to know the water quality in the different containers well, that water was also analyzed for the nitrates, and phosphates. So here are the results of the river water analysis. From the 9th to the 11th of November, it was still possible to apply fertilizers to the fields, whereas the samples taken from the 15th to the 17th of November were taken during the fertilizer ban period. It was found that the average before phosphate concentration is 0.15 milligram per liter lower than the average after phosphate concentration. The before average nitrate concentration is 2.5 milligram per liter higher than the after average nitrate concentration. I was unable to detect nitrate, probably, while that doesn't mean that that there weren't any, but probably the concentrations were just too low for the aquarium test, which were then not sensitive enough. And samples from the water protection areas, so the sample from the Lac La Haute-Sur, only shows residual concentrations of the measured ions. And to evaluate the effect of mania on aquatic microorganisms, I used the hemocytometer to estimate the abundance and size of the different microorganisms, and the results are shown on the left in the table. The nitrate, and phosphate concentrations in the different containers are shown on the right in the diagram. Results showed that in the containers with no or little slurry, we could find mainly microorganisms that could be described as large or very large. On the contrary, in the containers with several milliliters of slurry, there were mainly microorganisms that could be classified as small or very small. These classifications are quite random. However, the microorganisms classified as very small are in the order of magnitude of bacteria. So thus, there is a bacterial proliferation if the water contains a lot of animal detritus, while non-bacterial aquatic microorganisms die. To conclude, in terms of river water quality, it can be said that in periods with a fertilizer ban, the nitrate concentrations decrease. The water in the water protection zones, so the water of the Lac de la Haute-Sur, has the lowest concentration of nitrates and phosphate measured. In terms of impact of mania on aquatic microorganisms, it can be said that the populations evolve differently in water of different quality. Water with a lot of organic detritus kills non-bacterial microorganisms while it stimulates bacterial proliferation. In summary, therefore, my research has confirmed that regulation of fertilizer use has a real impact on water quality and aquatic population. So this was the project I participated in the Jonk Fuerscherin competition with. Shortly before the competition itself, Luxembourg came to its pandemic-induced stop and the jury sessions were switched online. And thankfully, I was very honored to be awarded the participation prize at the European Union Contest for Young Scientists, Sport for Youths in Salamanca, Spain. It is organized by the European Commission, and it is the biggest and most prestigious science competition in the EU. Initially planned to happen on September 2020, it was cancelled, so we couldn't travel to Spain, sadly, but we had a very beautiful alternative prize ceremony. And last year, the UCS committee announced that they were organizing a combined UCS where they were combining the UCS 2020 and UCS 2021 under a hybrid version. So the Spanish contestants were in Salamanca, while the international contestants participated via an online platform, and the contestants from 2020, so including me, could participate nevertheless. And while online contests aren't exactly the same experience as contests in real life, at least it was made possible that we could have an international contest with intercultural exchanges and meeting other scientists working in the same field is a very enriching experience and a lot of fun. So participating in the Jonk Fuerscherin contest has taught me a lot, and there are especially 3 things I wanted to share with my fellow young scientists. The first lesson learned was that when signing up for the contest, one doesn't have to be an expert in the chosen field. One doesn't have to be a great scientist either. But getting started on a fascinating subject leads us that way. In the process, we learn how this scientific method works, how a paper is structured, how to defend the project in front of the jury, how to explain it to other people and to share it with the world. And to give an example, at the beginning of the contest I participated in, I didn't know that the theoretical information belongs in the introduction part of the project paper instead of the interpretation part. But I learned it along the way, and that's what matters. And a second lesson I learned, and which I find very important as well, the water analysis were made with aquarium tests. They were brought online at a very low cost. The microscope and the hemocytometer were lent by my school. So you don't necessarily need a fully equipped laboratory to do good research. Because with quite basic means, I did get results I could work with. The aquarium tests weren't able to detect nitrates, that was true, but they did detect nitrates and phosphates. I did get results to work with. So doing science is really accessible to everyone. And finally, last lesson I learned was to try to see the strong points of our project. Because when working on a project, one does make mistakes, or at least I did. And we also always seek to improve our projects. And because we know them so well, and we also know the weaker points of it. This gives us the opportunity to work on them, that's right. But the risk is that we kind of miss the strong ones. And that would be sad, because we also have to defend our project in front of the jury. And while defending it, we have to point the strong points out. And so don't miss these, and try to be proud of yourself. Thank you.